Good afternoon, everybody. I'm pretty sure that most of you must have heard that the heavy metal lead is contained in these daily products. But are you aware of the health impacts that it brings to our body? Lead attacks almost all systems in our body. For example, in the nervous system, it can cause brain damage. While in the cardiovascular system, it can cause anemia. And in the renal system, it can cause kidney failure. Despite its severe impacts, most people ignore and neglect the presence of lead in our daily products. For instance, 30% of primary students in Jakarta and 90% of homeless students in Makassar are lead poisoned. And 80% of toys manufactured in China contain 4 times more lead than the national standard allowance. This matter, however, is not responded seriously by our government. Then what should we do? Should we just wait for the solution to be proposed by our government, or can we propose our own solution? A research was conducted by these researchers, which is investigating the purification of water by using brown algae. But why in water? Why should we try to control 70% of the whole Earth's surface area, where we can control something that is closer to us, something that is inside us? Why not in lead? It still solves the problem, but it's easier to control, and it's definitely not as much as all the water sources in our world. And for these reasons, we conducted a research entitled The Inhibitory Effect of Sargassum on Blood Level. My name is Dominicus Untariady. I'm representing my partner, Dominicus Untariady. We are from Santa Lorenzo. Why do we choose sargassum? There are basically 2 reasons to this choice, which is first, the abundance of sargassum, especially in Indonesia, and second, its ability to bind heavy metals. This ability is caused by the alginate content in their cell walls. This molecule possesses the carboxyl functional group, which can be used to form complex ions by forming hard-covalent bond with a metal ion, which in this case is the lead ion. And this mechanism of sargassum binding the lead ion is called by absorption. Lead itself interferes and enters our body through 3 pathways: through inhalation, ingestion, and skin absorption. After it has entered our body, it will be circulated in our blood, either bound to the protein hemoglobin or exist as free ions. The main function of hemoglobin is to bind oxygen. However, it can also bind lead due to the presence of the carboxyl functional group. And if lead keeps on entering our body, our blood lead level will increase, and if it exceeds 10 micrograms per deciliter, it is considered as lead poisoning. One of the mechanisms that were disturbed by lead is the hemoglobin synthesis. It interferes with the ALAD enzyme, causing the low rate of erythrocyte production and the short lifespan of the erythrocytes produced. In this research, we would like to measure the bioabsorbent effectiveness based on the biomass dosage and the contact time. The effectiveness itself is defined by measuring the decrease in blood lead level, erythrocyte count, and the hemoglobin concentration. In order for us to achieve these objectives, we divide our research into 2 phases. The first phase is the in vitro experiment in water, in which we would like to determine the equilibrium state of the bioabsorption against lead. So the variable that we use is the contact time. While the second phase of our experiment is the in vivo experiment in rats. This phase is conducted to prove the applicability of this bioshaping technique in living beings, and also at the same time examine the ability of sargassum as both cure and prevention toward lead poisoning. So we use the variable biomass dosage. We started our first phase by adding standard sargassum to a solution of lead nitrate, in which this solution are agitated in the water shaker, where we adjust the condition to be similar to blood's condition. The duration of the agitation is 1 to 12 hours and 24 hours. And after it is done, we centrifuged it for 15 minutes and analyzed it using the atomic absorption spectroscopy to know the final concentration of lead remaining in the solution. The data of this phase can be seen from this screen. This graph shows the amount of lead bound in the biosorbent calculated by using this formula. As you can see, the biosorption rate increases until it has reached a point around 50 milligrams per gram at a time of 3 hours. Starting from this point, the biosorption rate stays constant, which means it has reached its equilibrium state. We can explain this phenomenon by the fact that by this time all the carboxyl functional group being used up for binding the lead, so there are no further lead binding. Well, the second phase of our experiment is begun by giving conditioning time to the rats for 7 days by giving them rat pellets. After the conditioning time is over, we gave 8 kinds of treatments to these rats. The first group does not receive any treatment in particular because this is the control group. The second group received only the lead treatment. This is done to simulate the condition where lead poisoning is not cured. The next 3 groups are given lead after being treated by 3 dosages of sargassum. This is to portray the function of sargassum as a prevention to lead poisoning. While the last 3 groups are given 3 dosages of sargassum after being treated by lead. It is conducted to portray the function of sargassum as a cure to lead poisoning. After all the treatments have been given, we analyzed the blood samples from each rat by using 3 tests, which are spectrophotometry to know the concentration of hemoglobin, the atomic absorption spectroscopy to know the final blood level, and also the hemostometry test to know the amount of erythrocytes in the blood. Then there comes another question. Where will the lead go after they are bound by the biosorbent? To answer this question, we are going to conduct 2 tests, which are gas chromatography for the fur, to know the deposit of lead in the fur, and the atomic absorption spectroscopy for the urine, to know whether lead could be excreted to our urine. We have reached the point of the conditioning stage and is about to begin the treatment stage. So up to this point, we can conclude that sargassum works well as a biosorbent against lead, and it will reach its equilibrium state, which is around 50 milligrams of lead per gram biomass by the time of 3 hours. John Wooden once said, "It's the little details that are vital. Little things make big things happen." We don't expect that this research would be an absolute solution to this problem, but what we are trying to achieve is to raise the awareness to severe yet neglected disease, so we can bring a healthier and higher quality of life to this world. Thank you.